So for chapter 9, we are going to be looking at the patterns of inheritance. And this is expanding on what you have learned about genetics so far. So dogs are one of man's longest genetic experiments. Over thousands of years, humans have chosen and mated dogs with specific traits. It is not, I think it's common knowledge now that the modern dog is derived from the wolf. So a domesticated wolf befriended humans and were then raised by humans, and specific traits were chosen or desired. And so the result has been an incredibly diverse array of dogs, all with distinct body types and behavioral traits that are seen today. So you can see things from like the Great Danes to the Chihuahuas and everything in between. So for this video, we're going to be looking at Mendel's laws. We're going to be looking at the history of genetics and Mendel's introduction, his contribution to genetics, and his laws that he has added and discovered and helped create the modern Mendelian genetics that we know today. So the science of genetics has ancient roots. It can be traced as far back with the pangenesis proposed around 400 BCE by Hippocrates. This was an early explanation for inheritance that suggested that you inherited particles called pangenes that came from all parts of the organism that were incorporated into eggs and sperm, and that these characteristics could be acquired during the parent's lifetime, could also be transferred to the offspring. While it was I think it was a great idea at the time. I mean, 400 BC is a very long time ago. Aristotle did reject the pangenesis idea and argued that instead of particles, it was the potential to produce the traits that was inherited. So the idea that hereditary materials mix in form an offspring, called the blending process, was then proposed around the 19th century by scientists studying plants. But this was later rejected because it did not explain how traits that disappear in one generation could then reappear in later generations. So where does our modern understanding of heredity and genetics come from? It comes from Gregor Mendel. Heredity is the transmission of traits from one generation to the next, and genetics is the scientific study of heredity. Gregor Mendel spearheaded this field of genetics in the 1860s, and he deduced the principles of genetics by breeding garden peas, and then relied upon a background of mathematics, physics, and chemistry to help him come up with his laws for genetic distribution and variability. In 1866, Mendel correctly argued that parents pass on to their offspring discrete heritable factors. At the time, Mendel didn't know exactly what these heritable factors were. He just knew that there was something that was being passed from the parents to the offspring. And he stressed that the heritable factors retained their individuality generation after generation. So we now know today that these heritable factors are called genes, and your genes are passed from your parents to the offspring. So a heritable feature that varies among individuals For example, flower color is called a character. It's a characteristic of the flower. Each variant for a character, such as purple or white flowers, is a trait. Um, so you can have, for all the many different characteristics that a species or organism may have, there can be many variations or many different traits of those characteristics. This is a picture of Gregor Mendel, the monk from the Abbey. So one of the, in my opinion, one of the most brilliant things that Mendel did was the idea of creating true breeding varieties. And so true breeding varieties are the result of when you self-fertilize that plant that produces offspring that are all identical to the parent plant. So, for example, if you have a plant that has white flowers and you will self-fertilize from that white flower, and all of the offspring are identical to the parent plant in that they have white flowers, then you have created what they call a true breeding variety. The offspring of two different varieties are called hybrids. So, for example, if you have a white flower and a red flower and they produce an offspring, you could produce a hybrid offspring that now has pink flowers. The cross fertilization is a hybridization or genetic cross. So whenever time you cross any two individuals, you are creating a genetic cross. True breeding parents are the P generation. They're always designated as the P generation. The hybrid offspring are the F1 generation. And then across of those first generations, you can produce second generation plants, labeled as F2 generations. So this here is just your everyday garden pea. You can see that it's got this is a diagram of the flower, showing you that it's showing you the sexual organs of the flower there, where the carpel is the female organ, and then the stamens here are the male organ of the flower. And so Mendel was able to self-fertilize and create true breeding plants or true breeding organisms by taking the pollen from this stamen and fertilizing the carpel here, which is usually not done in nature. It's not as common as you would think, but Mendel was able to do that. So this is just giving you an example of how Mendel, in his meticulous study of the pea plants, was able to pollinate different parental plants. And he's showing you he's taking the stamens from one individual, taking the pollen grains and adding them to the carpel here, the female organ of another individual. So he's cross-pollinating these two parental plants that will produce offspring. So this is the fruit from these cross of these plants. And these seeds will grow and develop into offspring. This is your F1 generation here. And you can see they are all purple, even though one of the parents had white color. So this is just a list of all of the different traits that Mendel looked at. He looked at flower color, flower position, seed color, seed shape, pod color, stem length. And then he looked and he determined that some traits seem to be occur more often than other traits, and that some traits seem to be more dominant than other traits. Mendel's law of segregation describes the inheritance of a single characteristic. So a cross between two individuals differing in a single character is what we call a monohybrid cross. Mono meaning one, hybrid meaning hybrid individuals, and the cross between them. Mendel performed a monohybrid cross between a plant with purple flowers and a plant with white flowers. The F1 generation, so the offspring of the parental cross, produced all plants with purple flowers. And so then when he made a cross of the F1 plants, they produced an F2 generation where 3/4 of them had purple flowers and 1/4 of them had white flowers. And so this is just the visual representation of that experiment where you have two true breeding parental plants, true breed purple, true breed white, which when they're mated or crossed with each other, their offspring were all purple flowers. So the F1 generation was all purple. But then when you mate or cross two F1 generation plants, the offspring though had a ratio where 3/4 of them were purple and 1/4 were white. So the all purple F1 generation did not produce light purple flowers as predicted by the blending hypothesis that was proposed in the early 19th century. And so Mendel needed to come up with a reason why you didn't have light purple flowers. Why did all of a sudden a white flower pop back up in the F2 generation? So white colors seemed to disappear, but then it reappeared in the F2. So Mendel observed this same pattern of inheritance for 6 other pea plant characteristics. So Mendel developed from this experiment these 4 hypotheses, which are going to be now rewritten and used modern technology. 1, he recognized there had to be different versions of those heritable factors, which we now know as genes. Different versions of genes are called alleles. Different alleles account for the variations in inherited characters that we see in many of the characteristics and traits that we observe. For each characteristic, an organism inherits 2 alleles, one from each parent. The alleles can be the same or they can be different. So if you are a homozygous genotype for that trait, it means you have identical alleles. Homo meaning one, zygous meaning it has to do with the zygote characteristic. So one type of allele, they are identical compared to someone who could be heterozygous, which means they have 2 different alleles for that trait. 3, if the alleles of an inherited pair differ, then one determines the organism's appearance, and it's called the dominant allele. The other has no noticeable effect on the organism's appearance, and it's called the recessive allele. So the phenotype is the appearance or expression of a trait, and then the genotype is the actual genetic makeup of a trait. So a phenotype may be determined by more than one genotype. So let's think of something, an example. An example would be color. Anything color related has multiple, it's a multiple gene trait. So the phenotype would be the actual appearance or color And then the genotype would be the genes that are there, the genetic makeup. It's the actual nucleotides of the DNA that makes up that type, that gene, and then that trait. And then the fourth hypothesis that he proposed was that a sperm or egg carries only one allele for each inherited character because allele pairs separate or segregate from each other during the production of gametes. And so it is that statement that is called the law of segregation, in that you only inherit one allele from your parental gamete. Mendel's hypotheses, it also explained the 3-to-1 ratio in the F2 generation. In the F1 generation, all the hybrids had a one dominant, one recessive genotype, allele genotype. So you can use a Punnett square that can show the possible combinations of alleles that could occur when gametes combine. So this is the true breeding parental generation, where you have true breeding purple, which has 2 dominant alleles, compared to white flowers, which have 2 recessive alleles. And so when you cross these 2 true breeding parental generations, you will create an F1 generation that is now all hybrids. It's a true hybrid of the parental plants, in that they have one dominant and one recessive allele. So all of the plants, all of the organisms here in the F1 generations are always hybrids. But when you cross these, when you cross the F1 individuals cross hybrids for F2 generation, so if you take the sperm from one plant, you'll have one dominant allele, one recessive allele. And then if you take the egg from another plant, one dominant allele, one recessive allele, and you cross them. So you take this dominant will create this offspring individual. Or this dominant and this recessive will create this offspring individual. And you'll notice if you keep doing that and you fill in your Punnett square, you will get one individual that is completely what we call homozygous dominant, has 2 dominant alleles, 2 individuals that have one dominant, one recessive allele, and then 1 individual that has both recessive alleles. So if you're looking at the phenotype, the physical characteristics of these traits, then 3 of them are purple and 1 of them are white. But if you look at the genotypic ratio, you'll see it is in a 1-to-1 ratio, where you have 1 completely dominant, 2 that are hybrids, and 1 that is completely recessive. And this is just reiterating that same concept that I just went over. It's important to note that homologous chromosomes bear the alleles for each character that is observed. So a locus, remember a locus, we talked about the locus in the last chapter, is a specific location on the gene, the specific location of a gene along the chromosome. For a pair of homologous chromosomes, alleles of a gene reside at the same locus. That's so a lot of time and research has been spent to isolate and detect specific genes, and finding them on one chromosome, you can find them now on the homolog of that chromosome. Homozygous individuals have the same allele on both homologs, where heterozygous individuals have a different allele on each homolog. So this is just a representation of two homologous chromosomes. They have not been replicated, so they don't have the traditional X shape that you were accustomed to in the last chapter. These are just the two homologous chromosomes that are there. One is the parental, uh, one is the maternal chromosomes that you inherited from your parents, for example. And so on each chromosome here, you have loci that are labeled, and it's showing you if they are dominant or recessive. Um, Capital letters mean they are dominant. So here is the genotype, two capital letters for this allele, and then it's homozygous dominant for this allele. And then here's an example where you have two recessive um, alleles because they're two lowercase letters. So this is homozygous for the recessive allele. And then here's an example of something that is heterozygous in that it contains one dominant allele and one recessive allele for this character or trait.